So last time was really just a primer to give you guys an idea of what genetics is. So now we're going to start digging into the science, and we're going to first look at the cell. So if you guys have ever taken high school biology, you're probably very familiar with what a cell is. But as a refresher, it's important to remember that all of life is made up of cells. So anything considered living has to be made up or is a cell. So for something like skin cells, so the cells that make for skin, if you zoom in close enough with a microscope, you can actually see the individual cells that make up our skin. So here in this microscope picture, you can see the individual cells. Here they're actually stained with a pink dye, so you can actually see them under the microscope. So humans are actually considered multicellular. So just as the name implies, we're made up of multiple cells. So humans have about 1 trillion cells. So a very, very large number of cells compose us. Similarly, plants as well are multicellular. So they're made up of many, many different cells. And here again, we have another microphotograph taken as a microscope picture. So you actually see the individual cells that make up the plant. So as opposed to being multicellular, we also have organisms that are made up of a single cell. So sometimes called unicellular, or more simply, single cell. So bacteria is actually a very good example of a single-celled organism. So as you know, bacteria are very small. So as opposed to humans and plants, which are very big in comparison to bacteria, bacteria are made up of a single cell, so they're very, very tiny in comparison. So again, all of life is made up of cells. So for something to be considered living, either has to have cells or be composed of cells, or it has to be a cell itself, as in the case of a bacterium or other microscopic organisms. So really today we're going to be focusing on the cell itself. So understanding how the cell functions as well as how they replicate, so how we make more cells. So as you guys can imagine in genetics, everything's happening at the microscopic level, so at the level of the cell. So to understand genetics, we need to understand how the cell functions. So that will be our main focus for today. And here's this just again for you guys in written form. So the cell is essentially the smallest unit of life. So we can't get anything smaller than that. So we're actually going to see that many cells have smaller components inside of them. So those components themselves of the cell are not considered living. The cell itself, including all the components that make it up, is considered a living thing. As we discussed, humans are made up of multiple cells, so we are multicellular, which is a very important word to know. Plants as well are also multicellular. However, microscopic organisms like yeast, bacteria, those are examples of single-celled or unicellular organisms. It's also important to note that especially in multicellular organisms, certain cells can take on special characteristics. So we have special cells that make up the liver, we have cells that make up the skin, we have muscle cells. So when cells are part of a multicellular organism, they can start taking on unique roles. When we get later into this course, we're going to discuss how those cells actually function. So what makes a skin cell, what makes a liver cell. So what's controlling genetics behind how different types of cells function. So that'll be a focus later in this course. So I've alluded a little bit to this, but cells you can think of essentially being tiny factories. So again, a cell is a living thing. So just like humans have a digestive system to break down nutrients, cells also have their own way of doing that. So they have special places where they can actually break down certain nutrients, and that's in what's called the lysosome. Similarly, just like the human has a brain, a cell has a nucleus where it stores information. So you can think of a cell having a lot of similar functions to the human body in a way. And a little bit later in this lecture, we're discussing the specific parts of the cell that actually do each of those functions and break it down piece by piece. But essentially, a major point here is to think of the cell as being a living thing. So imagine what you would need to do to be a living organism. So humans have to eat, we have to store information, we have to move around. Similarly, cells have a way of doing that, and we'll be discussing exactly how they do that later in this lecture. So before we actually talk about the specific components of the cell, let's talk about the molecular components first. So here we're zooming way in, even smaller than the cell. So we're looking at the very basic chemistry behind life. And it turns out that there are only 4 major different molecules or components that make up the cell. Here I use the word components, but I should be saying molecules. So if you haven't taken chemistry in a while, I'll go over what a molecule is in the next slide. You can imagine cells being made up of 4 different molecules. And when you think about it, we actually eat cells. So a lot of our diet, essentially 100% almost of our diet, is composed of these 4 major components or molecules. So for something like this hamburger right here, this is made up of the 4 major molecules we find in all of life. And as we're going to see, these 4 major molecules are found in all types of cells. So now let's actually figure out what those 4 components are. So those 4 molecules that make up all of life. And just as I said, we eat cells. So cells utilize these 4 molecules. So if we looked at something like the nutrition facts, we actually start seeing what those 4 major molecules are. So here again, we're looking at the nutrition facts. Here for one hamburger, it has 750 calories. So as we're going to see, a lot of the major molecules of life provide energy for the cell or for the organism. And here that energy is represented in calories. So now let's actually look at the components of this hamburger right here. So you can see we have fats, we have something called saturated fat, and we have cholesterol. So these 3 together are actually called lipids. So fats and cholesterols together we call lipids. So that's our first major type of molecule we're going to be talking about. The second major molecule are carbohydrates. So that's our second major molecule. You can see that we have various types of carbohydrates. We have dietary fibers. So these are indigestible plant fibers. So a lot of plant material we can't digest, we can't break down the carbohydrates, or essentially sugars in that plant material. And because of that, it just passes through our digestive system as dietary fiber. What you're probably more familiar with is something like table sugar or starch. So carbohydrates really, you can think of being sugars. But it's also important to remember that carbohydrates can also be things like dietary fiber. So plants actually have sugar molecules providing structure there. Number 3 is proteins. So something like the meat of the hamburger has quite a few proteins in it. So meat is made up of muscle cells, and muscle cells actually function by use of proteins. So as we discussed in lecture 1, we saw that proteins have many functions, and one of them is being muscle cells, or essentially being the contractile fibers that allow muscle cells to contract. So number 4 is actually not on the nutrition facts. So number 4 is actually nucleic acids. So nucleic acids are things like DNA and RNA. So here we don't actually list it on the nutrition facts. So it actually turns out that humans actually don't need to consume that much DNA or RNA in their diet. So the human body is very good at conserving DNA and RNA, so it's not something that our body normally gets rid of. So I believe from our diet, we only uptake about 1% of all the DNA and RNA we actually eat. So the amount we need is very negligible compared to these other 3 molecules we need. So lipids, carbohydrates, and proteins, we need much more of those than we do DNA or RNA. So there's one thing from these nutrition facts I actually skipped, and this is the sodium. So you can think of table salt. So while table salt is very important for the human body itself, it's not actually a major molecular component of the cell. So it's not something that we're going to be talking very much about. So it's really lipids, carbohydrates, proteins, and nucleic acids that make up a cell. So we've been talking a little bit about molecules, so I'm using the word quite a bit. So let's actually discuss what a molecule is. So a molecule is made up of multiple atoms linked together. And specifically, these atoms need to be of different elements, so different types. Okay, so now what's an atom? So an atom is essentially the smallest unit of matter. So just like a cell is a small unit of life, an atom is the smallest unit of matter, at least for our purposes. If you take quantum physics, you'll learn that there are actually things that make up atoms. So you can imagine something like a table is made up of atoms, a couch is made up of atoms, the air that surrounds you is made up of atoms, everything that's matter. So everything we can touch, we can feel, everything that exists is essentially going to be made up of atoms. And as I sort of alluded to, there are different types of atoms. Different types we call elements. So you guys have probably heard of carbon. That's a very good example of a type of element. Silicon is a type of element. Iron is an element, so on and so on. So just to review, we have a molecule. So a molecule is multiple types of atoms linked together. We then have atoms. So anything that's physical is going to be made up of atoms. Then we have elements, so different types of atoms. So these atoms have different behaviors. So a carbon atom is going to behave differently from an iron atom. So those different types of atoms we call elements. So these are really rudimentary definitions. If you ever take more advanced chemistry, you'll go into much more detail about each of these definitions. But here, this is what you need to know for the genetics we're going to be covering in this course. So now let's actually look at this molecule of guanine. So you'll become very familiar with guanine. So it's one of the common bases found in DNA. So as I said, this is the molecule of guanine. So here it's made up of multiple different types of atoms or elements. So we have things like nitrogen, carbon, hydrogen, and oxygen. So as you can see, we have abbreviated names of elements here. So N stands for nitrogen, C for carbon, O for oxygen, H for hydrogen. So you can see this molecule of guanine is made up of those 4 different types of atoms or 4 different elements. And if you look in between each of those letters, we have these lines right here. So these lines actually represent what we call a covalent bond. And we won't talk too much about what a covalent bond is just yet, so in later lecture we'll actually cover a little bit more about the types of bonds in molecules. But here, these covalent bonds are linking together different atoms. So here we have a covalent bond linking hydrogen to nitrogen. And you can see we have bonds throughout this molecule. So here we have another covalent bond. Here's another one. Here's another one. So remember, a molecule has multiple atoms linked together, and these have to be different types of atoms. So here we have hydrogen, nitrogen, carbon, and oxygen linked together in a various pattern. So here we have satisfied the definition of a molecule. So as we discussed, there are many different types of elements. So iron, silicon, titanium. So really all the symbols and all the abbreviations, all the information you need to do chemistry, or most of it at least, can be found in the periodic table. We won't actually have to look too much at the periodic table in this class though. So just like life is made of 4 major molecules, so lipids, carbohydrates, proteins, and nucleic acids, life is also made up of 4 major elements. So carbon, which is C, oxygen O, N for nitrogen, and H for hydrogen. So you're going to see those 4 elements come up quite a bit. So for exam purposes, memorize these 4 abbreviations. These are all straightforward. So carbon starts with a C, oxygen starts with an O, nitrogen starts with an N, and hydrogen starts with an H. So we're going to see these 4 elements come up quite a bit in this course, so it's important that you know them. And here's a periodic table of elements. So you can see we have quite a few different ones. So you have things like boron, which is B, copper, which is Cu, iron is actually Fe, so not I. So if you ever don't know what an element is, you can come here and figure out what's its abbreviation. So now we're actually going to discuss those 4 major molecules that we mentioned a little bit earlier in this lecture. So remember, those are lipids, carbohydrates, proteins, and nucleic acids. So the first one we're going to focus on are carbohydrates. So carbohydrates, you can think of things like sugar. So here we have some table sugar in the top left-hand corner of the slide. So that's a good example of a carbohydrate. On the right-hand side of the slide, we have 2 examples of carbohydrates. So here we have glucose. On the bottom, we have fructose. So all of you are probably familiar with those. So glucose is a very common type of sugar, fructose as well. So the more technical term for carbohydrates are saccharides. So a saccharide is essentially one single individual molecule of a carbohydrate. So here we're looking at monosaccharides. So here we have one molecule of glucose and one molecule of fructose. So of course, mono means one. If you link 2 molecules or 2 saccharides together, we call it a disaccharide. So essentially 2 sugars linked. You can do 3 saccharide molecules or 3 sugar molecules linked together. So that would be a trisaccharide. You can keep on going beyond that if you want to. Really, after disaccharide, though, we skip to polysaccharide. So poly means many. So here we have many sugar molecules linked together. So it's not very specific. But beyond disaccharide, most people just go directly to polysaccharide. We'll see the prefix poly come up again a little bit later in this lecture, so it's important that you're aware of what poly means. Looking at the functions of carbohydrates, they can be used for energy. So for something like a monosaccharide or disaccharide, those are used for quick energy. So if you ever drink a sugary drink, so if you ever drink soda or something like that has lots of fructose in it, that's going to provide your body with lots and lots of sugar to quickly use up. So that's why you usually get a sugar rush if you drink too much soda or if you eat too much candy or something like that. So lots and lots of mono and disaccharides. It's also important to note that carbohydrates can also be used for long-term energy storage. So polysaccharides are usually used for long-term energy storage. So as mentioned a little bit earlier, plants can also use carbohydrates for structure. So that's dietary fiber. Next, we have lipids. So butter is a very good example of a lipid, and butter specifically is made up of triglycerides, or fats, as they're commonly known. So lipids are hard to define, but they're always going to be hydrophobic. So oil, just like butter, is a very good example of a lipid. So hydrophobic is essentially a way of saying it does not like water. So hydro, you can think of water. Phobia or phobic, you can think scared. So here, lipid molecules are scared of water. So if you ever mix oil and water together, they separate into two layers. And that's because oil is hydrophobic, so it's scared of water and they don't mix together. So lipids are also a very diverse group of molecules. So there are three common types of lipids. Those are the triglycerides. So again, the fats. We have the phospholipids. So phospholipids actually make up the cell membrane. We'll talk a little bit more about this, but if you remember back to those cells I showed you at the beginning of this lecture, the outside of the cell is essentially covered with phospholipids. And lastly, we have steroids. So these are sometimes called sterols or cholesterols, depending on the textbook you're using. So steroids can be more common things to think of, like testosterone or estrogen are very good examples of steroids. But steroids can also be things like the cholesterol we get from our diet. So that isn't really for signaling, so that has a different function in the human body. So as I alluded to, steroids are very important for signaling. So these are hormones. Again, testosterone, estrogen, estradiol. Those are all examples of steroids. But again, things like the cholesterol we get from a diet, that is not an example of a signaling hormone. Triglycerides, so fats, as you can imagine, just like carbohydrates, are going to be used for energy. And the phospholipids, again, make up the cell membrane, which we'll talk a little bit more about later in this lecture. So next we have proteins. So as we saw with carbohydrates, which are made up of saccharides, here proteins are made up of amino acids. So when somebody is taking protein powder or something like that, essentially they're just eating a bunch of amino acids. Similarly with a hamburger, the meat of the hamburger has lots of amino acids, so lots of protein that can be broken down into amino acids. In life, there are about 21 common amino acids. So in the next slide, I'll actually show you what the 21 common amino acids look like. But the number 21 is going to be important when we do some calculation a little bit later on in this lecture. When we link a lot of amino acids together, so you can think of the amino acids being the building blocks of proteins, when you link a lot of those together, we make a polypeptide. So again, poly means many, and peptide relates to the bond that holds together amino acids. So polypeptide is essentially a bunch of amino acids linked together. When those amino acids get a 3D shape, we form a protein. So a protein is a polypeptide with 3D shape. So as with the first lecture, we talked a little bit about some of the functions of proteins. So in general, we say they do work. So these are usually globular proteins. So globular, you can think of like a glob. So globular proteins you can think of being sort of a ball or a sphere, so a glob of something. This is in opposition to a fibrous protein. So fibrous protein are mostly for structure. So these are things like hair. Hair is a very good example of a protein that's fibrous. So we have globular proteins, which usually do work, and we have fibrous proteins, which are usually for structure. We also have things called enzymes. So enzymes do certain functions in the body. So they also do work, but here we're going to be a little more specific. So enzymes also do work, but they're a little bit special of a type of protein. Later on in this course, we'll go a little bit into more detail about what enzymes are, but here know they do certain functions of the body, some of which we discussed in the very first lecture of this course. And just like the proteins that do work, enzymes are also globular proteins. And of course, as we saw on the first day of the course, proteins have many, many functions beyond what I've written here. On the right-hand side of the slide, I have an example of amino acid. So again, here we have a molecule. This is one amino acid. So there are really four major parts, 3 or 4 depending on how you want to split it, major parts of an amino acid. So here we have nitrogen, So remember, N is an abbreviation for nitrogen, and 2 hydrogens. So this is the amino group. On the right-hand side of this molecule, we have the carboxyl group. So here we have a carbon with bonds to oxygen and hydrogen, and then carbon with another bond to an oxygen. So this is a carboxyl group. We have the central carbon, and then we have the R group. So the R group is sometimes called the variable group. So these 4 parts make up an amino acid. So all 21 amino acids have these 4 parts. So they have an amino group, a central carbon, carboxyl group, and the variable group. So as we're going to see, the R group or variable group is really what makes amino acids different from each other. So here we have a table showing the 21 common amino acids. This is absolutely not something you have to memorize, but I want to show you some of the diversity we have in amino acids. So let's look first at arginine. So if you look closely, arginine has 2 abbreviations. So arginine can be abbreviated ARG, or it can be abbreviated with a single letter R. So again, you don't need to memorize this, but it's important to be aware if you see R. So if you see R by itself or ARG, they're referring to arginine. So again, let's look at those 3 major parts that we discussed. So here we actually have the carboxyl group. We have the amino group right here, a central carbon, and all of this right here is the R group, or the variable region. Next, we'll look at histidine. So HIS, or H, is histidine. So again, we have the carboxyl group, amino group, central carbon, and the R group. We can then look at lysine, LYS or K, carboxyl group right there, amino group, central carbon, and the R group. So if you look at arginine, histidine, and lysine, you can see the only thing that changes between them is the R group or variable region. So just as it's implied, the variable region is what's different between amino acids. So all amino acids have an amino group, a carboxyl group, and the central carbon. What's changing is the R group. Same idea with aspartic acid, or D, glutamic acid E, so on and so on. We can look at all these amino acids. The only thing that's changing is the variable group. Also, in regards to some of the chemistry here, it's important to note that every time you see a bend in a line like this without a symbol, this means there's a carbon there. So this is a short way of writing carbon. So whenever you see a bend in a line, that means there's a carbon there. So that's opposed to writing all those C's out. So again, we have 21 common amino acids. And what's changing is the variable region in each of these. So again, when we link these 21 amino acids together, we form a polypeptide. And when that polypeptide gets a 3D shape, we form a protein. So here we have an example of a protein. So here we have hemoglobin. So as you can see, hemoglobin has a nice 3D shape. It has 4 parts to it. And here we have essentially a protein. A critical thing to understand is that the 3D structure of a protein determines its function. So if a protein had a different structure than hemoglobin, it would have a different function. And when you consider this, you can start to understand how we can have proteins with many, many different functions. So if a protein had 100 amino acids, so if it was 100 amino acids linked together, we would have 21 to the 100 amino acid combinations. So even if you're not good at math or exponents, you're probably well aware that 21 to the 100 is an enormous number. So there are an enormous amount of different types of combinations of amino acids. And this is assuming that a protein is only 100 amino acids long. Some proteins can be much, much longer than that. So there can be many, many different combinations there. So just like we'd have 21 to the 100 different amino acid combinations, we would expect about 21 to the 100 different 3D structures. And again, the 3D structure determines the protein's function. So 21 to the 100 different 3D structures means 21 to the 100 different functions. So this is why I can't just simply give you a list of the functions of proteins. So unlike lipids and carbohydrates, which are mostly just about energy, proteins I can't give you such a simple definition of what they do. And that's because there are so many different functions, because there are so many different 3D structures. So proteins are extremely diverse in their functions. So lastly, we're going to talk about nucleic acids. Essentially, when you think of nucleic acids, you can think of DNA and RNA. So those are examples of nucleic acids. So nucleic acids are actually made up of three major parts. You have a phosphate group. So P is the abbreviation for phosphate. We have a sugar. That sugar is going to be either deoxyribose or ribose. So deoxyribose in DNA and ribose in RNA. And lastly, we have the base. So the phosphate group and the deoxyribose or ribose is always constant. So those never change. What is changing is the base. So as you guys know, DNA functions in heredity. So it codes for proteins. So the information is essentially stored in the base. So the information is essentially stored in the base. So just like in amino acids, as we saw with the variable region or the R group, you can think of the base of DNA and RNA being essentially the variable region. So that's what's changing in between the various nucleic acids. And this brings us back to the central dogma. So remember, we go from DNA to RNA to protein. DNA to RNA is through transcription. RNA to protein is through translation. So the central dogma, again, is sort of the underlying principle behind genetics. We're going to see it's not absolutely true later in this course, but here it's going to frame a lot of the discussions we're going to be having in the lectures. So as I mentioned in the last slide, DNA codes for proteins, and proteins have many different functions. So again, DNA is information, and through transcription and translation, we make proteins which have a certain function. And that function of the protein is determined by the 3D shape of the protein, which is ultimately determined by the amino acid sequence. So that protein function is encoded or stored in DNA. And again, we're going from genotype to phenotype. So we've been talking quite a bit about the chemistry behind the cell, but now let's actually talk about how a cell works. So here we're looking at an animal cell. So I'll preface this by saying that there really isn't a general animal cell. So we talked about how the human is made up of different cell types, so like kidney cells, muscle cells, liver cells. So each of those cell types is going to be a little bit different on the inside. So even though we're looking at a quote-unquote animal cell here, all cells are slightly different from each other. So don't think that every cell you're going to see is going to look exactly like this. So anticipate there's going to be some difference between those cells. But here, just to take a reductionist approach, we have a general animal cell that goes over a lot of the structures we need to talk about. So a lot of the structures within the cell are called organelles. So organelles literally means tiny organs. So as we've talked about earlier in this lecture, the cell has a lot of things that are analogous to human organs. So we're going to be talking about the peroxisome and lysosome, so both of those are involved with digesting food. And peroxisomes and lysosomes are both considered organelles. So now we're going to talk about the organelles that make up a cell. So organelle literally means tiny organ. So again, thinking of a human, humans have a lot of organs that process and do various functions in the human body. Similarly, organelles serve various functions within the cell. So the right-hand part of the slide, we have a cell with all of its organelles labeled, or at least the majority of them. So we're going to start with very outside the cell, and we're going to look at the plasma membrane. So the plasma membrane you can think of being the wall of the cell. So it keeps things from entering the cell, and it keeps things from leaking outside of the cell. So you can think of the plasma membrane being the wall of the cell. So essentially it's keeping things within and also preventing things from getting inside. In the next few slides, we'll actually cover a little bit more detail of the plasma membrane. So we'll talk a little bit more about how it functions, since this is in particular very important. So we started with outside, and now we're going to go from the outside to the very center of the cell. So usually in the center of the cell, you will have the nucleus. So the nucleus contains the information on how to make proteins within the cell. So the nucleus is actually enclosed by what's called the nuclear envelope. So you can think of the plasma membrane being for the whole entire cell. The nuclear envelope is specifically for the nucleus. So just like the plasma membrane is the wall of the cell, the nuclear envelope is the wall of the nucleus. And the nuclear envelope, essentially, as far as its chemical composition, is mostly lipids. So it's a lipid membrane, but it also has proteins lining the pores. So the pores are essentially holes in which information can pass inside and outside of the nucleus. Of course, importantly, in the nucleus, we also have the chromatin. So this is DNA plus associated proteins. So as you guys know, the nucleus is where you find DNA. But there are also proteins in there as well, and specifically proteins that associate with DNA. In later lectures, we'll actually identify those proteins and talk about their function. Within the nucleus itself, there's usually a darker dot or darker region within the nucleus. This is called the nucleolus. So this is a site of ribosome formation. So this is actually where a lot of the ribosome is constructed. So moving outward from the nucleus, we have the endoplasmic reticulum, sometimes abbreviated the ER. It comes in two versions. There's rough. Rough is in particular important for genetics. We'll talk a little bit about this in later slides. Smooth, though, so smooth ER, makes lipids. And these names are very intuitive. So if you look at the rough ER, you see it has this rough shape. So lots and lots of dots sort of lining outside of that. And those dots are due to the ribosomes that are bound to that endoplasmic reticulum. And you can see the rough ER is important for making secretory and membrane proteins. So remember, we go from DNA to RNA to protein. That's our central dogma. So any protein that needs to either leave the cell or be embedded into the plasma membrane has to go through the endoplasmic reticulum, specifically the rough ER. Smooth ER, as you would expect, is smooth. So that's this stuff right here. And it's very important for making lipids and steroids. Now let's look at the Golgi apparatus. So the Golgi apparatus sort of has a stack of pancakes look. So you can see there's multiple stacks on top of each other. So that's a very good sign that you're looking at the Golgi apparatus. And the Golgi apparatus is all about modifying proteins. So as we're actually going to see in later slides of this lecture, some proteins are modified. So they actually have things like sugars added, sometimes lipids, and various other molecules added to them. So the Golgi apparatus is all about adding these extra molecules. So no longer you just have amino acids, you can also have amino acids, various other molecules attached to them. Next, we have the mitochondria. So the mitochondria you can think of maybe being like the stomach or something like that. So the mitochondria is all about producing energy. And it has this very characteristic bean shape. And you can see there are little walls or protrusions all throughout. So the mitochondria is all about producing energy. An interesting fact about the mitochondria is it has its own DNA. So even though the cell stores most of its information in the nucleus, there is some information stored within the mitochondria. So this is a very interesting fact because we're going to learn a little bit about mitochondrial genetic diseases. So there are actually specific genetic diseases that are due to changes in DNA within the mitochondria. So it's important to know that mitochondria also has its own DNA. Not nearly as much as the nucleus, but still enough that changes within that DNA can have a huge impact on the human body and human disease. Next, we have peroxisomes and lysosomes. You can also think of these being similar to the stomach. So peroxisomes and lysosomes are fairly similar. They have this sort of circle shape. They normally fuse with some sort of vacuole, though. So the vacuole you can think of being almost like a container. So if the cell has swallowed something, so imagine it's swallowed some food or something floating around, that vacuole is then going to fuse with the peroxisome or lysosome. So the peroxisome and lysosome have lots and lots of digestive enzymes or digestive proteins that break down food. The peroxisome in particular is for breaking down lipids. Not 100%, but in particular, lipids. And the lysosome digests essentially everything else. So again, the vacuole itself is sort of like the stomach. The peroxisome and lysosome are then the digestive enzymes or the holder of digestive enzymes. So those fuse with the vacuole and you have your food digested. Those digested particles can then go onto the mitochondria and be converted into energy. So all of this stuff so far, or everything in the cell really, is floating around what's called the cytoplasm. So the cytoplasm you can think of being sort of the inside of the cell. So cytoplasm is made of what's called cytosol. So the cytoplasm is the actual space. The cytosol is what actually makes up the cytoplasm. So the cytosol is mainly just water, but it's also the stuff that's floating around in it. So the proteins, lipids, and whatnot that are floating around in the cell. So lastly, we have the cytoskeleton. So even though it's called the skeleton of the cell, perhaps thinking of it more as our cardiovascular system is probably a better analogy. So the cytoskeleton is mostly about moving things around the cell. So even though we're looking at a picture right here, which is static, it's not moving, a lot of these organelles are capable of moving. So you may see the mitochondria moving, you may see certain proteins, even the nucleus itself can also move. And the cytoskeleton plays a huge role in that. So there are various types of cytoskeletal proteins. So you have microtubules, intermediate filaments, and microfilaments. I don't expect you to know too much detail about the cytoskeleton, but be aware that the cytoskeleton has these specific functions. We'll also see how the cytoskeleton plays a huge role in how cells divide. So in regards to this image right here, it's very important that you look over the learning objectives so you understand what you need to learn from this and what you need to be able to do. Definitely at the very least, I expect you to know what each of these organelles do, but I also expect you to be able to identify them when I show you a picture of a cell. So a lot of this will be covered in the practice we will be doing in the next lecture. So now we're going to go over a couple of key structures, so structures that are in particular important for genetics. So first, we're going to talk about the plasma membrane. So the plasma membrane is made up of lipid molecules, and specifically phospholipids. So if you remember back to a little bit earlier in this lecture, there are three major types of lipid molecules: triglycerides, steroids, and we have phospholipids. So phospholipids are the major component of the plasma membrane. You can see we have a phospholipid molecule right here. And if you look closely, the plasma membrane is not just lipid molecules or phospholipid molecules. It's also made up of lots and lots of proteins. So the plasma membrane is not just simply a wall. There are lots and lots of functions that it's able to do, thanks in part to these proteins. So these proteins do things like sense the environment, transport molecules, and many more functions beyond that. You also see that there are many different types of proteins. So you have things like surface proteins. So here we have the cytoplasm of the cell. So this is on the inside. And on the outside, you have the extracellular fluid. So essentially what's on the outside of the cell. So you see we have surface proteins on the inside. We have integral proteins. These are proteins that span the whole entire plasma membrane. We have protein channels to transport proteins. And as we discussed, proteins can also be modified. So here we have a glycoprotein. So glyco, you can think of glycogen or glucose, so essentially carbohydrates. So here we have a protein that has some sort of sugar molecule added to it. And this was done by the Golgi apparatus, which we talked about 2 slides ago. Next, we have the nucleus. So again, the storage site of DNA, so a very important organelle to know. So I won't go into too much more detail than what we've covered in the last few slides. As you can see, we have the nuclear envelope. So you can think of that being almost like the plasma membrane of the nucleus. We have pores within that nuclear envelope. So this allows for information to enter and leave the nucleus. We have the nucleoplasm. So just like the cytoplasm, so essentially the space within the cell, the nucleoplasm is the space within the nucleus. We have chromatin, so our DNA plus protein. We have our nucleolus, which is that sort of darker dot right here. So the site of ribosome formation. And then we have the endoplasmic reticulum. So the endoplasmic reticulum is actually separate from the nucleus, but as we saw, is very important for secreted and embedded proteins. So proteins that need to leave the cell or proteins that need to be embedded into the plasma membrane. So here's actually what the rough endoplasmic reticulum actually looks like within a cell. So you can see all this stuff right here, that is the rough endoplasmic reticulum. So you can see it has a lot of these dots everywhere. It does have that very rough shape. But it's important to remember the endoplasmic reticulum comes in both rough and smooth varieties. So rough is all about secreted and embedded proteins. The smooth variety is all about producing lipid molecules. And as we discussed, the reason why the rough ER is rough is because it has lots and lots of ribosomes docked to it. And the ribosome is a critical structure for genetics. So it translates or does translation. So it takes RNA and creates proteins off of that. So you don't need to know too much about the ribosome at this point, but know it's made up of 2 major parts. You have the large subunit and small subunit. So those are the 2 major parts of the ribosome. And the ribosome, as I said, is all about translating RNA into amino acids or proteins. So this lecture is really all about the cell. So it's chemical composition, its organelles, and now we're going to talk about how it actually makes new cells. So if you think about life, a critical thing about it is that everything needs to be able to reproduce. And similarly, cells need to be able to reproduce. So this last portion of the lecture, we're going to be covering what's called mitosis and meiosis, essentially how we make new cells, or in the case of meiosis, how we make sperm and egg. So here's really the big picture. Some organisms need to do asexual reproduction, and all organisms need to grow. So asexual reproduction, you can think of a starfish. So if you cut the arm off of a starfish, or if the starfish loses an arm, it can regrow a whole entire new starfish from that arm. So this is asexual reproduction. So that starfish doesn't have to mate with another organism. If it just lops off its arm, that arm is able to regrow into a whole entire new starfish. So those 2 starfishes are essentially going to be clones of each other. And as you all know, all organisms have to grow. And this specifically is in regards to multicellular organisms. So all multicellular organisms grow throughout their lifespan. So asexual reproduction and growth is all due to the process of mitosis. So mitosis is all about creating identical cells. So as I said with the starfish, you have clones of each other. So mitosis creates identical cells. For sexual reproduction though, so reproduction where you have mating, or essentially sperm and egg meeting, you go through the process of meiosis. So in meiosis, you create sperm and egg, and you want variation. So you don't want each sperm and egg to be exactly the same. You want them to be slightly different. And this variation is all due to the fact that we want genetic diversity. So genetic diversity usually increases the chance of an offspring living. So variation is actually a helpful thing. So variation is actually helpful when it comes to sexual reproduction. And here we have a baby chick, which I think illustrates both those concepts very nicely. So here we have an egg. So that baby chick was formed from the fusion of the sperm and egg. So the egg from the mother and the sperm from the father met, and that made a fertilized embryo. The chick itself, though, is going to go through the process of mitosis. So through the process of meiosis, we had formation of the egg. And the sperm. Through the process of mitosis, we're going to have the chick grow up into an adult chicken. In general, though, mitosis and meiosis are just simply organized movements of DNA. So essentially in mitosis and meiosis, we're just moving DNA around. But as you can imagine, this has to be a very, very precise process. If it's not precise, we start getting genetic abnormalities. And this can cause a lot of issues. So that's why these movements of DNA have to be very, very precise. So we don't want too much DNA in one cell and too little in the other. Again, this is going to cause lots and lots of issues. In regards to the end products of mitosis and meiosis, in mitosis we produce two cells. So we start with one cell, produce two. In meiosis we produce 4 daughter cells. So we start with one cell in meiosis and we produce 4 sperm or 4 eggs. That's not exactly true. When we talk about eggs, actually, we only produce 1 egg from one cell. The 3 other cells become what are called polar bodies. But for now, just for simplicity's sake, know that in meiosis, we start with 1 cell, we end up with 4 sperm or 4 egg. We'll go into more detail about that once we do reproduction. So now we're going to talk more about how DNA is actually stored within the nucleus. And this is just a simple reminder to show you that DNA is in fact stored within the nucleus. So here we have a stain that marks DNA. This is probably what's called DAPI. So it's a stain that binds to DNA and it fluoresces. So as you see, this blue color right here is the nucleus of the cell. And you can see all that blue stuff is our DNA. If you look on the far left, though, we actually have a cell that's probably undergoing mitosis. So this cell right here is undergoing mitosis. So you actually see the DNA sort of clumping together, and the DNA is actually moving within the nucleus. So again, mitosis and meiosis are essentially just movements of DNA within the cell. So you guys probably have a conception that DNA is essentially just this double helix right here, and that's how DNA exists within the cell. So it's not entirely accurate. So if DNA was in this form right here, the form you're very familiar with, it's just plain double helix, it would stretch about 3 meters long. So that's for a human cell. So clearly 3 meters is not going to fit within a cell. So DNA is usually bundled up, and this is due in part to these proteins called histones. So you can think of histones sort of coiling up DNA as wrapping it tighter and tighter. So here you can actually see the DNA wrapping around, so it's become a tighter coil, tighter and tighter. So DNA is not always like this though. There's a gradient between packed very tightly and loose. So some DNA is loose, so some DNA is like this, and some DNA is like this. We'll talk more about in later lectures why exactly this is. But it turns out that loose DNA is more active. So if a gene is very active, you're going to have loose DNA. If a gene is not very active, you're going to have it tightly packed. So essentially stored away for later use. So again, these histones are all about packing DNA. Eventually, if DNA continues to pack further and further, we can get very, very condensed pieces of DNA. So something like this right here. And so this tightly packed DNA, I like to think of being like balls of yarn. So nice balls of yarn like this. So remember, mitosis and meiosis is all about moving DNA. So you want something very nice and compact to move around. You don't want something like this pile of yarn right here. It's going to be a lot harder to move that around. So that pile of yarn is just like loose DNA. So you want those histone proteins to start condensing DNA to make it tighter, packed tighter and tighter, and create these balls of yarn right here so you can have them move around very nicely during mitosis and meiosis. And all of this is happening in the nucleus. So this is a very dynamic process. So you actually have histone proteins raveling and unraveling DNA constantly. So it's always changing. So it's a very dynamic process. So don't think it's something that's static like it is in this picture. So for DNA, it's usually kept in what is called chromosomes. So we don't have just one long piece of DNA within our nucleus. We actually have 46 smaller pieces called chromosomes. So right here we have one chromosome, here's another one, here's another. So our DNA essentially is kept on 46 different strands. And you can notice that some of those strands are longer, some of those strands are shorter. So something like 21 is a lot shorter than number 1 right here. So notice that all of these chromosomes are paired together with another chromosome. So you can see we have 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, and 2 number 5s. So the reason why we pair chromosomes is because one comes from your dad and one comes from your mother. So for chromosome 4, we have one from your dad and one from your mother. Same idea with chromosome 5, we have one from dad, one from mother, same as 6, 7, 8, onward and onward. So all of your chromosomes, you have 23 from your dad and 23 from your mother for a total of 46. So we'll come back to this idea that you have half your chromosomes and half your chromosomes from dad and half your chromosomes from mom a little bit later in this lecture when we talk about homologous chromosomes. So what we've just gone over is called a karyotype. So karyotype essentially shows all the chromosomes within one of your cells. So it can be very helpful for diagnosing certain genetic diseases. So something like Down syndrome, so trisomy 21, that's very easy to diagnose with a karyotype. So if this was a trisomy 21 karyotype, you would notice it would have a third chromosome 21 right here. But in general, humans have 46 chromosomes, 23 from dad and 23 from mom. So as you can imagine, mitosis and meiosis is a very complex process for a cell. So to create a whole entire another one of your cells is very, very complex and requires a lot of planning for the cell. And because of that, it's a very tightly regulated process. And this process is regulated through the cell cycle. When something bad happens, when the cell is not behaving according to the cell cycle, you start to see diseases like cancer come into play. And this is due to the fact that the cell is not dividing properly, so it's not going through the process of the cell cycle correctly. So you can think of the cell cycle almost being like a clock for the cell. So there are 4 major parts. There's G1, S, G2, and then the mitotic phase, or sometimes just called the M phase. So G1 and G2 are just all about cell growth. So those are phases where the cell is just checking to make sure it's large enough, so the nucleus is large enough, the mitochondria is large enough. So you're going to be splitting the cell essentially in half. So the cell wants to make sure it's large enough to do that and not totally destroy itself during the process. The S phase, you can think of S standing for synthesis. So the S phase is all about DNA synthesis. So we want essentially 2 of everything at the very least for a cell. And for most cells, you're only going to have 1 copy of its DNA. So for each human cell, you usually only have 46 chromosomes in each of your cells. During S phase, though, you have to duplicate all of that. So here you need 92 chromosomes to get ready for mitosis or meiosis. So during S phase, you have synthesis or replication of DNA. And this replication or synthesis of DNA is a very critical process that we'll actually be covering in a later lecture. So we'll have a whole entire lecture talking about how exactly the cell makes more DNA. Together, G1, S, and G2 are called interphase. So essentially the whole entire gray region, so from here to here, that is interphase. So G1, S, and G2 are interphase of the cell cycle. In M phase, that's actually where we have mitosis happening. So in M phase, we actually have formation of 2 new cells. So that really is actually quite a quick process. So if you think of this pie chart being time, you can see the cell spends most of its time in G1, S, and G2 phase. So M phase is actually very, very short. And that's because mitosis and meiosis are all just movements of DNA. So moving DNA does not take quite as long as synthesizing it does, as well as growing the cell. So it's a very short process in comparison to interphase. So in addition to G1, S, and G2, there's also something called G0. So for cells that are not dividing, they can go into G0 phase. So you can think of G0 almost being like exiting the cell cycle. So the cell cycle implies that all cells are going to be dividing at some point. G0 is for cells that are not going to be dividing too much. So a lot of the cells that make the heart or the nervous system, a lot of those are probably going to be in G0 phase, so they're not going to be dividing too often. So again, G0 phase is for cells that are not going to be dividing. But be aware that you see there's an arrow coming back in, so cells can enter and exit the cell cycle depending on where they are in their life cycle. So now let's look at DNA replication. So here this is going to be a little more detail, but not quite as much as we're going to be covering in later lectures. So this slide is mostly just about terminology. So we talked about how we had 23 chromosomes from dad and 23 chromosomes from mother. So we call these pairs homologous chromosomes. So if we go back to our karyotype, here we have homologous chromosomes, homologous chromosomes, homologous chromosomes. So one from dad, one from mother. So during S phase of the cell cycle, we're going to have replication of these homologous chromosomes. So dad's chromosome is going to replicate and mom's chromosome is going to replicate. So now we have sister chromatids. So we have sister chromatids right here and right here. So you can see each of those homologous chromosomes, one from dad, one from mother, each of those replicated. So now we have essentially 2 chromosomes stuck together. And these 2 chromosomes stuck together we call sister chromatids. However, all of these together are still considered homologous chromosomes. So don't confuse sister chromatids with homologous chromosomes. So homologous chromosomes states that we have 23 from dad, 23 from mother. Sister chromatids are what occur when we have replicated chromosomes. And eventually those will split during the process of mitosis, and now we'll have one cell right here and another cell right here. So we'll have 2 cells formed during the process of mitosis. So again, we can go over this on the right-hand side of the slide. So here we have homologous chromosomes. So all 4 of those together are homologous chromosomes. Here we have sister chromatids. And here again, we have sister chromatids. So sister chromatids are replicated chromosomes. Homologous chromosomes are one from your dad, one from your mother. And really what makes something considered a homologous chromosome is that you have similar genes from dad as you do from mother. So looking closely, and so here's an example of unreplicated homologous chromosomes. So just as I pointed out, these are unreplicated. Replicated chromosomes usually have an X shape. So as we saw in the previous picture, they have two chromosomes lined up next to each other. So these two sister chromatids lined up have this sort of X shape right here. So here we have homologous chromosomes that are unreplicated. So finally, let's actually cover the process of mitosis. So remember, mitosis is all about creating two identical cells. So it's mostly for growth and asexual reproduction. So here are the 5 or 6 major steps that a cell undergoes during mitosis, or M phase. So the first thing that happens is we have prophase. So in prophase, the chromosomes condense and become visible. So remember back to histones and DNA. So remember DNA is bound to histones. So as histones start winding DNA tighter and tighter, so it's making those nice compact bundles of yarn so that the chromosomes are easier to move around. So the fancy way of saying that is that the chromosomes condense. We also have formation of spindle fibers. So these things right here. So these are actually part of the cytoskeleton. So this is mostly going to be about moving around the chromosomes. So you can think of the spindle fibers attaching the chromosomes and moving things around. So those start becoming visible during prophase. We also see the nuclear envelope breakdown, as well as a nucleolus disappearing. So essentially we're getting rid of things in the way, so we're breaking down the nuclear envelope, we're allowing the spindle fibers to actually attach to the chromosome to start moving things around. So we don't want the nuclear envelope there, since that's going to get in the way, to prevent those chromosomes from moving around freely. So at the bottom we actually have the rest of the microscope picture showing this happening. So in the green we actually have the spindle fibers. In the blue we have the nucleus, or essentially the DNA that makes up the nucleus. In prometaphase, so the second step of mitosis, chromosomes continue to condense, so they continue to pack tighter. We also have what are called kinetochores forming at the centromeres. So this is probably very confusing, so we don't talk too much about this yet. But the centromere you can think of being essentially the center of the chromosome. So we go back. Right here, this is the centromere. So centromere is essentially the center of the chromosome. The kinetochore is where the spindle fiber is actually going to attach to. So the kinetochore is actually some protein that the spindle fiber can attach to. And that's the third step here. So we have mitotic spindles attached to the kinetochores, and the centrosomes themselves actually move towards opposite poles of the cell. So you can see we have one on the left and one on the right. So essentially they're moving to opposite sides of the cell. And that's exactly what we see here. So you can see we have the spindle fibers at either sides of the cell. So that's because the centrosomes move to other sides. And we also have the DNA becoming more condensed in the middle. In metaphase, so the third step, we have chromosomes line up on the metaphase plate. So you can see that the sister chromatids have lined up nicely in the center, and that's really the major way to identify the step of mitosis. So the chromosomes line up on the metaphase plate. Also, the mitotic spindle is completely developed, and we have all sister chromatids attached to the spindle fiber. So you can see we have the metaphase plate right here, so all that DNA is lined up, and we have the spindle fibers on either side of the cell attached to each of the sister chromatids. Next, we have anaphase. So you can think of anaphase being like apart. So we have the sister chromatids actually separated now. So sister chromatids are pulled apart. So you can see we have one set over here and one set over there. And more appropriately, these sister chromatids are no longer sister chromatids. They're now just called chromosomes. Next, we have telophase. So telophase is actually sort of the end of mitosis. So we start to see the nuclear envelope come back. That's a major difference between anaphase and telophase. So we see the nuclear envelope come back. We also see reduction of the spindle fibers. And we also start to see the chromosomes decondense, so they become less compact. And finally, remember, the whole entire point of doing mitosis is to create two new cells. So through the process of cytokinesis, which isn't really a step of mitosis, we actually have the cell split into two. So all that's going to happen is you have a pinching of the cell together. So this space right here is going to be pinched together, and you're going to have two new cells created. So this is cytokinesis. So now we're going to focus on meiosis. So mitosis is all about creating identical cells. So if you imagine we started with one cell, it had 46 chromosomes. In mitosis, we're going to create two cells, each with 46 chromosomes. So we're creating identical cells. Meiosis, though, is considered a reduction division. So if we start with 46 chromosomes, we're going to end up with 23 chromosomes in each cell. Specifically, we're going to have 4 cells with 23 chromosomes each. So 4 sperm or egg with 23 chromosomes. And hopefully this makes intuitive sense to you. So a normal human has 46 chromosomes. So when a sperm with 23 chromosomes meets with an egg with 23 chromosomes, we get a normal human with 46 chromosomes. If we had 2 cells with 46 chromosomes meet, we would get 92 chromosomes, which would be very bad. So for sperm and egg, they have 23 chromosomes each, so half the normal number of chromosomes, so that when they meet together, 23 plus 23 makes 46, or a normal human cell. So really, mitosis and meiosis are almost exactly the same thing. There's some very subtle differences you need to be aware of, though. I'll be covering a little bit of that in this lecture, and also in the practice you'll be doing in the next one. So really, the first thing that happens is interphase. So this wasn't shown in the figure for mitosis, but really the first thing that happens in mitosis is you have interphase. So remember, if you go back to the cell cycle, interphase is G1, S, and G2. So we have cell growth and, very importantly, DNA synthesis. So before mitosis, so before prophase, we have interphase happening, so we have replication of all the chromosomes. Similarly, in meiosis, we have all the chromosomes duplicated. After interphase, we have meiosis 1. So meiosis is split into two major parts, meiosis 1 and meiosis 2. However, they follow the same steps as mitosis. So we have prophase, prometaphase, metaphase, anaphase, telophase 1, and cytokinesis. So prophase 1 is very similar to prophase and mitosis. So we have breakdown of the nuclear envelope. We have formation of the spindle. The major difference, though, is we have formation of what are called tetrads. So you can see that we actually have chromosomes lining up on top of each other. So specifically, we have homologous chromosomes binding firmly together. So sort of sitting on top of each other, forming these tetrads. So tetra means 4. So you can think of 4 chromosomes, actually 2 pairs of sister chromatids, sort of like 4 total, sitting on top of each other and forming this tetrad. So during prophase 1, we have this very important process called crossing over happening. Exactly what crossing over is, I'll talk a little bit about later in this lecture. But know that in prophase 1, we have formation of tetrads and crossing over happening. So this is different from mitosis, where that does not happen at all. Prometaphase 1 is very similar to prometaphase and mitosis, so we just have chromosomes lining up. Metaphase 1, though, is very different from metaphase and mitosis. So if we go back to metaphase and mitosis, you see we have a single line, and I'll actually delete what I've drawn here. So you can see we have a single line of sister chromatids lined up. This is very different from metaphase 1 of meiosis. So instead of sister chromatids lined up, here we have homologous pairs lined up side by side. So this is a major difference between metaphase 1 and metaphase of mitosis. In anaphase 1, since we've had homologous pairs line up instead of sister chromatids line up in a single file row, here we have homologous chromosomes separating. In anaphase of mitosis, we have sister chromatids being pulled apart. So again, anaphase of mitosis, we have sister chromatids pulled apart. Anaphase 1 of meiosis, we have homologous chromosomes pulled apart. Telophase 1 of cytokinesis of meiosis 1, though, is very similar to mitosis. So we start to see the nuclear envelope come back. And we have 2 cells formed during cytokinesis. So all of that together is considered meiosis 1. Again, there are 2 major parts of meiosis, meiosis 1 and meiosis 2. So essentially, we're going to have the whole entire process happening again. So in prophase 2, we're going to have breakdown of the nuclear envelope. It's very important to know that there's no crossing over. So crossing over only happens in prophase 1. And again, what crossing over is, we'll talk a little bit later. We'll talk about that a little bit later in the lecture. Prometaphase 2, we just have more development of the process. And now metaphase 2 is exactly similar to metaphase of mitosis. So now we're splitting, or now we're getting ready to split sister chromatids. So exactly the same idea as metaphase and mitosis. Similarly with anaphase 2, we have splitting of sister chromatids. So anaphase 1, homologous chromosomes separate. Anaphase 2, sister chromatids separate. And then telophase 2 in cytokinesis, we finally have 4 cells in total formed. And each of these 4 cells, again, have 23 chromosomes each. Half the number in a normal human cell. And I should say, essentially this process is happening twice. So remember in meiosis 1, we form 2 cells. So one cell is going through all these steps. Another one is also going through the same steps as well. So essentially, meiosis 2 happens twice. So really a major point of meiosis is trying to create variation. And this variation is usually good, so it's more likely that an offspring is going to survive if it has more variation from its mother or father. So variation is a good thing in meiosis. And if you understand the reason why cells undergo mitosis, you can understand that variation in mitosis is a bad thing. So the first way in which a cell can create variation during meiosis is through the process of crossing over. And this again occurs only in prophase 1, so not prophase 2. And this is exchange of alleles between homologous chromosomes. So we talked a little bit about alleles in the first lecture, but alleles, remember, are different versions of genes. So here we have the capital A version of a gene, and here we have the lowercase a version of a gene. So here you have a different version of gene A from your dad than you have from your mother. Also notice that we have 2 copies of each of these alleles. So you have 2 copies of the uppercase allele and 2 copies of the lowercase a allele. So remember, these are replicated homologous chromosomes. And because of that, we have 2 copies of each of these alleles. If it was unreplicated, it looks something more like this. You have A, B, C, A, B, C. So during crossing over, we actually have exchange of genetic material between homologous pairs. So you can see here we actually have information being exchanged between these chromosomes. And when this happens, we have recombinant chromosomes formed. So here we have chromosomes recombined into a slightly different configuration than what they started as. We also have non-recombinant chromosomes as well. So the chromosomes on the outside have not exchanged information. So now you can see we have genes A, B, and C all uppercase on the left, genes A, B, C all lowercase on the right. The chromosomes that have recombined, you can see we now have different alleles on the inside. So uppercase A, uppercase B, and lowercase c, and lowercase a, lowercase b, uppercase c. So now we created more genetic variation here. And I encourage you to actually go through the steps of meiosis, so actually draw them out and see what cells we can come up with due to this process of crossing over. So draw what would happen. So draw with crossing over. So what are the possibilities if you do have crossing over happening? And draw the possibilities without crossing over. And for the sake of simplicity, do this only with 2 alleles. So with 3 alleles, it can be quite complex. So imagine you only had 2 alleles. So imagine you got rid of C here. So here you only have A and B alleles. So what are the possible configurations due to crossing over? So this is a very helpful exercise that will help you understand why crossing over creates variation. The second way in which we can form variation during meiosis is independent assortment. And this is just the fact that chromosomes can align differently during metaphase 1. So remember, during metaphase 1, we have lining up of homologous pairs. So we have one homologous pair here, and a second here. One here, and one here. So notice that the homologous pairs can line up differently. So here's one possibility, and here's a second possibility. And this is assuming we have 4 pairs of sister chromatids. So depending on how these are aligned at metaphase 1, they'll be pulled apart differently. So these will pull apart to the left and pulled apart to the right, pulled apart to the left, and pulled apart to the right. And this all affects eventually what you get at the very end of meiosis. So if you have this configuration, these are your possibilities. If you have this configuration, these are the 4 possible cells you can get. So due to independent assortment, so essentially aligning it differently at the metaphase 1 plate, you can have various combinations of cells created. So just like I had you guys do the exercise with crossing over, I highly recommend you do the exercise again, this time with independent assortment. So imagine what would happen if you had independent assortment and what would happen if you didn't have it. And also I recommend drawing on alleles to help you think about this process.